So we do these two that don't have the ampicillin so that we can see if the cells are actually alive. Because what we have here is E. coli that we put with DNA, so it has the plasmid in it, and we wanted to see whether it would pick up the plasmid or not and whether it would grow at all. It would be alive. This one doesn't have DNA, and the reason we did both of these, if they're both pretty much the same, is so that you could see whether the DNA was harmful or not, or whether it needed DNA to live. So if this one didn't grow, you would see that the DNA was harmful. And if this one...
Can you tilt them this way a little bit?
DNA. No DNA. Ooh. Sarah?
And then this one just doesn't have the DNA on the ampicillin plate. So you can see that there's a clear difference between these two, of course. And so without the DNA, you don't have the
resistance to ampicillin.
Thank you.
These plates are also loaded with ampicillin. So if the cells don't pick up the plasmid, they die, because the plasmid also codes for ampicillin resistance. So basically, all these cells have died. But if they didn't die, we would know that the cells were already resistant when we grew them, or they mutated a resistance, which is why we do all 4 of these plates and not just the one we want to look at, which happens to be kind of cool. This one has 2 different colors. It has a plasmid that codes for blue fluorescence and a plasmid that codes for green fluorescence, which is pretty cool. You can see really clearly the difference between the 2, even though the green shines brighter. That's how we know that the plasmid was picked up. Also because it's alive, mainly. This one is just blue. You can see it has a little bit of green. Probably there was some residue on the tip or something.
Or just more blue.
And this one's all green.
Super cool.
Super beef.
Transformation works.
Transformation works?
I love it.
Really cool.
Yeah,
Nice.